All right, guys, welcome back. So now we know how muscle contrast. Now we know these interaction between thin filaments and thick filaments. We are going to introduce the motor neurons into it. So the nerve, we have the motor neurons located in the spinal cord. It will send out, so it's kind of far, right? You can move your finger and the motor neuron is in the spinal cord. It will send out this axon to the muscle. Now, one motor neuron can control several muscle fiber. So you can see here, this is the nerve. The nerve ending has many axon branch, and that many axon branch will innervate. Each muscle is controlled by one nerve, but one nerve can control several muscles. So it's very much like a dog. You only have one owner, but one owner can have multiple dogs, right? So it's like that. The motor neuron can control multiple muscles. These one nerve control a group muscle. We call this a motor unit. The reason we call it a motor unit is because this is the finest movement you can conduct with motor neuron. So when we want to do any movement, you will only, the nerve will control a group muscle. And so when you excite that nerve, all those group of muscle will get excited all together. The motor units can be large. The benefit of getting a large motor neuron is that you can have a larger force. So one nerve can can control a lot of muscle together. It will generate a larger force. But with that one, you cannot have fine movement. And on the other hand, you can have a motor neuron to control small amount of muscle that will give you the fine movement, but you cannot have large force from those. So that's the motor unit. And so you know what's the motor unit? That's one nerve control many muscles. It's not. So that's that, right? Now, the nerve and the muscle, when they interact, it's just like nerve contact with the nerve. So this interaction is called synapse. Synapse represent the microscopic contact of two cells, but these two cells do not physically contact with each other. There is a gap, and this gap is, so we will need neurotransmitter release from one cell to the next cell. So that kind of like control the direction of the signal transduction from presynaptic side, which is an axon terminal, to the postsynaptic side. Before we talk about the postsynaptic side as a nerve, those postsynaptic side usually is a dendrite or soma. But here the postsynaptic side is on the muscle. So here is the skeletal muscle, here is that. So these synapse is called neuromuscular junction. All right. Yeah, so this is the term neuromuscular junction. Nice. Yeah, so that's how we call it, the neuromuscular junction. Why is it? It's the synapse from a motor neuron to a muscle cell. So that's that. So when people talk about why is a neuromuscular junction, neuromuscular junction is the motor neuron talk to or communicate with the skeletal muscle. So we have three type muscle. The other two type muscle, we don't have that junction. You use is the thing about the neuromuscular junction, you can see here. So it's a junction. It doesn't contact, but in a way, it kind of embedded into it. So that's how we improve or ensure that precise control the nerve release neurotransmitter, they gonna get spilled away. If it's not insert into it, it will get spilled away. So you're gonna excite several other muscles. So you don't wanna, so in order to improve the fine movement, the nerve getting deep into the muscle surface. So that's that. All right. So with this synapse, we need to know two things. Neurotransmitter. What is a neurotransmitter? The second thing is that what is a receptor? So neurotransmitter for neuromuscular junction is easy. It's only one thing. It's called acetylcholine. So here, the neurotransmitter released from the nerve end is called acetylcholine. So that's that. Quick question: what is the neurotransmitter of neuromuscular junction? Acetylcholine. Now, acetylcholine will bind on a receptor on the muscle. What is the receptor? This receptor is called nicotinic acetylcholine receptor. This is a ligand-gated sodium channel. So this is let me show you. Now we talk about several ion channel, right? Let me show you we talked about here in earlier slides. So let me just open it and I will just share it, stop sharing, and I will share it again to show you what we have learned. So it's here. So that's this one here, share. All right, you can see that here. So this is the one in a cell, and there are 4 types of the ion channel. Earlier, we talked about the voltage-gated ion channel, voltage-gated sodium channel, voltage-gated sodium potassium. So these ion channels are proteins. They anchor on the membrane, and they can be gated or non-gated. If it's non-gated, it's always open. We call this one a leakage channel. If it's gated, it can be gated by different way. It can be chemical gated. We call it a ligand gated, gated by a ligand, a chemical. Chemoreceptor is a ligand gated. Here is acetylcholine gated, so it's ligand gated. We also mechanically gated. This one will be sensitive to mechanical change, such as touch. Or baroreceptor, this is mechanically gated. But this is one ligand gated. Yeah, so that's that. Let me go back to these slides. So this nicotinic acetylcholine receptor is an example, is another member of ligand-gated ion channel. So is ligand-gated? What is that ligand? Interesting question. What is that ligand? Acetylcholine is the ligand, but nicotine can also activate it. That's why we call it a nicotinic acetylcholine receptor. Acetylcholine binds on it, causes it to open. Nicotine can bind on it to cause it open. Is this nicotine the smoking nicotine that people talk about? Yes. So that's the same nicotine. That's why we have this name. Nicotine can have effects in the brain. Its main effect is to induce arousal to make a person alert, awake. So if you're tired, smoking, that's why people smoke in the first place. They need to work overnight. It's just how we drink coffee now. So people smoke. But smoking, nicotine is very addictive. This is the strongest addictive. I don't know why people still allowed to smoke. It's very addictive. It's very difficult to, you got that excitement, but you got addicted to it. And so here, in our muscle, we have this receptor, nicotinic acetylcholine receptor. In our brain, we also have those receptors. So nicotine has stronger effects in the brain. Yeah. So that's that. So the ligand is now ligand-gated ion channel. Ion, what ion? All right. So in this case, nicotinic acetylcholine receptor, the ion is a sodium channel. So sodium can go in or out. It will open, sodium is freely move across. The net effect is from high concentration to low concentration. So in the cell, it is cell and this outside. So do you have more sodium inside of cell or outside of cell? As I have said this many, many times, the only thing high inside of cell is potassium. So sodium is not potassium. So sodium is high outside of cell. So when your gates open, sodium jump in, flow in. When sodium coming in, it bring positive charge ion. Sodiums are positive charge. Here you can see Na+. Positive charge, it will bring in, it will make the membrane potential to be positive. You excite it, you depolarize it, and then it will trigger an action potential of that muscle cell. The action potential will have depolarization, which is voltage gated. It's different channel. It's voltage gated sodium channel, and then voltage gated potassium channel have that action potential. So that's how we excite the muscle. Nerve excite muscle by acting on NACHR, nicotinic acetylcholine receptor, to cause sodium influx. And by injecting sodium into it, you move the membrane potential to be above the threshold and then to trigger an action potential. So that's that. All right, so that's here. Muscle excitation, axon terminal, here acetylcholine, action potential travel down to the axon terminal. These acetylcholine are saved in the vesicles. Acetylcholine release binds on nicotinic acetylcholine receptor, sodium influx, generate action potential, and the action potential will travel. It will travel throughout the cell membrane. Let me show you the cell membrane again. So this is cell membrane here. So this is a cell membrane. So action potential will travel along the cell membrane, right here, cell membrane. But muscle is thick, so this traveling will also get guided through the T-tubule into it. So it will still go to the cell membrane, but kind of near everywhere in the cell. And so that's how they excite the entire muscle. This action potential basically will spread throughout the cell membrane, well, spread through the T-tubule. And when they do that, they will cause calcium release. Calcium are stored in the sarcoplasmic reticulum. We have a lot of calcium stored here. They are not in the cytosol. Cytosol, the only thing high is potassium. So calcium is stored in the sarcoplasmic reticulum. So we are almost to connect the dots. We will have excitation, action potential travel through the T-tubule. You need to excite it, release neurotransmitter, bind on it, generate action potential, action potential travel through the T-tubule. T-tubules are nearing the sarcoplasmic reticulum. It will cause calcium get released. All right. So until then, you have a calcium release. Calcium release then binds on troponin, opens up, remove the tropomyosin, opens up, and then you will have myosin head binds on it. Then you have muscle contraction. So that's that. Is that good? All right. I hope that makes sense. So that's that. We talked about it. It's right here. So action potential travel down here. They will be very near the sarcoplasmic reticulum. Don't worry about this DHP receptor, etc. But all you need to know is that the action potential travels through the T-tubule. It will cause calcium release from where? That's a quick question. Calcium are stored in where? Sarcoplasmic reticulum. So, and then calcium get released. Calcium will then bind on troponin. Now it's blocked. You can see that. Myosin head is going to touch the actin, but not because tropomyosin blocked it. And then calcium binds binds on a troponin C or T-tubule again. T-tubule is an extension of the internal extension of the plasma membrane or cell membrane or sarcolemma. So that's an extension. And then it will cause the calcium flow out. Then calcium binds on troponin C. And then calcium binds on here. Calcium going out, binds on troponin C. Remove the tropomyosin. Actin is now exposed. Then myosin binds on it. ATP energize it, and then you have the sliding movement. The Z disc gonna move. So that is it. All right. I think that's everything. Yeah. So any questions? So here showing you that there is a delay between excitation and the contraction, because once you excited, it has to go through several steps in order to lead to the calcium outflux, and then you can have contraction. So that lead to a concept that is very commonly known, that we have fast twitching muscle and slow twitching muscle for different sports. You may want to train your muscle to be more fast twitching or slow twitching, or yeah, so different type of teaching use different type of primary energy in a way that some muscle use more stored energy, and some muscle use more of the oxidative phosphorylation right away, aerobic exercise basically. So that's that. So here is action potential generated by muscle by the NACHR, so the influx, and then you have action potential travel, then you have twitch. Yeah. All right. Any questions? Let me know. Okay. And that is all about the skeletal muscle. I will see you next time.